All right, masters of cell structure and function. Now it's time to step back and take a look at the organism as a whole for a moment and try to answer this question of how do organisms actually grow and get larger? Because if we recall, one of our characteristics of life was all organisms, whether you're multicellular or unicellular, have to grow and develop. So we're going to look at how they actually grow and get larger, and it's actually a pretty simple and easy concept. So remember that if we look at this, the cells of a blue whale the same size as the cells from an ant. And if we look here, two different skin cells here, these being from a blue whale, these being from an actual something small like an ant. I want to let you in on a secret, it's not really from an ant, but we'll pretend they are. And if we notice that they kind of look similar, they're about the same size, general characteristics and diversity. We can see a nucleus there. So the question begs, what makes a whale so massive and an ant so darn tiny? And the secret to this, what we want to focus on today, is actually cell division. And that's, we know that cells have to divide, and they do it quite often, some of them more so than others. And the real secret is that they can only get so big before they have to divide. Now here we can actually see these are a bunch of cells. Perhaps these are skin cells in a computer animation. And right here, it's a cell in the process of actually splitting in half. We can see these finger-like projections over on this side, some on this side, and eventually they'll become two identical cells. So it's our job, we want to look at how do they actually do that? What actually makes them divide? Because there are two main reasons as to why they have to divide. And in the first here we can see that this sounds a little nerdy, but it's actually pretty straightforward. It's a surface area ratio of surface area to volume becomes too big. What we mean is that as a cell grows larger and larger and larger and larger, it encounters a problem in that a cell this big and a cell this big. Now obviously we'll label them A and B. Cell B is much larger. However, the surface area, how much area on the outer surface obviously is bigger, but the volume actually increases at a faster rate, all the stuff inside of the cell. So as the cell gets bigger, it'll actually develop a problem in which it cannot exchange materials. Remember that most of these sugars that are going inside and the waste that's going out has to rely on diffusion across the membrane. And if a cell becomes too big, there's too much volume. And that presents a major problem. And in relation to that, we also have to think about and look at an idea of DNA overload. That DNA, remember, inside of our nucleus is what controls the cell. So eventually a cell could become so large it actually, the instructions, the DNA, the nucleus, and the boss from our last unit cannot actually handle all the requirements that are placed on the cell. So they have to divide. Now along those lines, one very important aspect, something you're probably familiar with, is the idea of chromosomes. And when we say, remember that DNA are the genetic instructions of the cell, that all from here on out, every time we see the term chromosome or we see these weird X-like structures, it literally is just DNA coiled up to form the structure. Because if we think back inside of our, looking at our cell, in our nucleus we can see a darkened spot. That really is where we keep our DNA. And remember that our DNA is actually for all intents and purposes, a loose, stringy, long strand of molecule full of instructions. And all that we do when a cell is ready to divide, it will actually take that DNA and coil it up so that it becomes a condensed body. In essence, really, it becomes this coiled up structure. There's our chromosome. So a chromosome is DNA is inside of our nucleus. And we need to, it's very important that we remember that idea as we progress forward. When we look at these things. And again, here's another example of that inside of our cell, our nucleus. During cell division, we can see chromosomes, and all a chromosome is coiled up DNA. Don't worry about what these letters are yet. We'll get there later in the year, but for now, DNA is a chromosome inside of our nucleus. So before we can keep going forward and look at how a cell actually divides, we've got to be concerned with its life cycle. And if you think of a human life cycle, you're born as an infant, you grow into a small toddler, then a child, then a tween, then a teenager, and a young adult, then an adult, then a middle grumpy age person, and so on until you die. Sounds pleasant, doesn't it? A cell's life cycle is a little bit different, and we break that up into parts and stages, consisting of G1, S, G2, and then M, which we're going to see is actually where the cell divides. And that's the end of the cell cycle. And it's rather than dying, usually what happens is it'll go back to G1, and the process will start over again. So if we look at this in terms of time, or almost like a clock, rather, in terms of time, we can see that at our initial point here, starting right here, as we progress through this way, here's this green eggs and ham, I do not like them, Sam I am, as our cell, and it'll go through G1, it'll pass through S, it'll pass through G2, and then finally this is where the cell actually divides, and what we're going to be concerned with, M, for the process of mitosis. And that's very, very important for us. So in looking at this a little bit closer, what actually goes on in this G1, S, G2 before a cell divides? We can see that most of the time is spent in G1, S, and G2 in interphase. The actual cell division, as we said, M, is rather short compared to the others. So very quickly, G1 referring to gap because it doesn't look like the cell is doing anything, when in fact it is. It's growing, it's doing its quote-unquote normal cell activities, making energy in the mitochondria, getting rid of waste, using lysosomes to break down damaged organelles, all of the things that we've been learning about the cells normally do. But they call it gap because it doesn't look visibly like the cell is doing anything. But this is a cell being a cell. Very important in the S, standing for synthesis. This is a big one. This is important because this is where our DNA, all of our instructions, are going to be copied. So we now actually have in our cell, after this phase is over, 2 complete copies of DNA. Bet you can't guess where each of those copies are going to go. Followed up with G2, gap 2. It looks like it's, again, like it's not doing anything, but this is where the cell is manufacturing energy, maneuvering organelles. It's getting ready for cell division. And then the actual M phase, mitosis, will go into those phases. That's where the cell will begin moving around its DNA and dividing. Very important. So one quick point before we look at the actual steps of mitosis is this idea of chromosomes. This can be a little bit confusing, so very, very important to take at least 1 or 2 notes here. And that is when during S phase, when DNA copies itself, and then later on we'll see they condense into chromosomes. They're actually not chromosomes yet. The reason it looks like an X is very, very important, and we want to look at this closely and make sure we realize that, what we're actually seeing here. Notice that we have put little colored markers here to represent genes. Genes are the directions, the instructions for, let's say, how to make, let's say this one is a gene for having blue eyes. Now, if we go and copy all of our DNA, when they condense into chromosomes, what will happen is that we have a copy for blue eyes here, and we also have a copy for blue eyes on the other arm. We can see that this arm, or this chromatid, as it's called here, is an exact replica of the other chromatid. That's why we call them sister chromatids. So this is actually a doubled copy. It'd be like having a copy of a book and then photocopying the other and sticking it on the end. 2 complete sets here, because what's going to happen is that one of these arms and all of its information here will go to one cell, one will go to another cell. So very important to remember that when we see this X structure, they are sister chromatids joined together with lots of genes on them. That one arm is exactly the same as another. Very, very, very important. And we do not want to forget that. That is super, super important. So, in order for the cell to divide, then how does it actually do it? It's really simple. It just takes practice being able to recognize them, which we'll do. Actually have to go through 4 to 5 phases here we're going to see. Prophase, metaphase, anaphase, telophase or telophase. PMAT are the 4 main stages before it actually divides. Now I've listed here, and you may wish to pause the video at this point to jot down briefly what's happening in each. I tried to summarize it in a sentence, the major event of what's occurring. What we want to do, though, is go through and look at each one of these phases up closely so we can see what's actually happening. So let's go ahead and do that. We don't want to watch the video here. We'll skip and do that in class. So for mitosis here, we're going from... So again, back with our interphase here, G1, S, and G2. and we can see all the rest of this is actual cell division. So we want to go through each one of the phases here and see what's actually occurring, look at them closely so that we can recognize them, so that we are able to see how a cell actually will go ahead and indeed will divide, because the process again is simple. So number 1, it's first in line prophase. And what I've tried to do is give you a cheesy little cartoon representation followed up with a real image here. And what we can see if we look at this, let's focus on the right hand side here, is that each one of these boxes is one cell. Here's our nucleus that we can see, and if we look inside, we can begin to notice some weird things. Now over here, these lightly colored guys, they're in interphase. We can't actually see anything. Really, we do see nucleoli inside of them. However, we don't really see much in there. It's just lightly shaded. There's all DNA in here. But here we can actually start to see distinct darkening spots. Now this is actually what's happening is our chromosomes are beginning to form, is the big idea. Our DNA begins condensing into chromosomes in prophase. So we're going to start to see some differences in what the cell begins to look like. Now it does look very much like interphase, and we got to be careful there, but they're darker. We can begin to see the actual chromosomes in prophase. This is very, very important. We can actually start to see some of our nuclear envelope beginning to break up as well, because that's beginning to deteriorate. We want to free up the chromosomes so that they can maneuver outward. So there's our prophase. That's an easy one. Moving on to metaphase is actually probably the easiest phase to recognize. All right, so as we said, metaphase will be a very, very easy, probably the easiest aspect of cell division to recognize, because in it, in our cartoon version, we can see all of our chromosomes have lined up single file at the center of the cell. So looking again at with a different stain, but a very, very similar set of cells. These are plant cells, remember? So here's one cell, as our cell wall, and in it we can actually see our chromosomes are bunched up and lined up in the center of the cell. They are lined up along the M middle of the plate. What's going to happen is we're going to separate these guys. One half of our copy will go to one end, the other half will go to the other end. But in metaphase, all they've done is our chromosomes have lined up at the middle of the cell. They're getting ready to move, to go to where they need to go. So this is an easy one. Meta, middle, no worries, no problems. Onward to anaphase is actually just as easy with the letter A, ana, away, or apart. And if you look at the cartoon image here, we can see that our chromatids, our copies, have been pulled to opposite ends of the cell. Remember here that this arm, this chromatid, is the exact same copy as this chromatid. If this has a gene for blue eyes, there's a gene for blue eyes there. Oh, a gene for having a big, ugly Bentley egghead? Oh, there's a gene for a big, ugly Bentley egghead. Exact copies. So, once again, looking at our awesome cell, oh fun, back to the opposite stain here. This is progressed a little bit farther along. All around us here we can see mostly a lot of interphases here, maybe going into prophase. But we can begin to see that there's one chromatid, there's another one. It's hard to pick out individual chromatids here, but they're being actually pulled to opposite ends of the cell. Now, when we say pulled, we've got to be aware of what's actually pulling these guys. You can very faintly see small little lines here. And those lines are actually, over here, are microtubules. They're large proteins that attach to the chromatids and help separate them. They actually help maneuver them all around the cells. So as they shorten, they decondense, depolymerize. That actually pulls our copies of chromatids to, in fact, opposite ends of the cell. So that's a good one as well, to be able to recognize what is happening there. So almost done, kids. We go on to telophase, and telophase is pretty easy. We now have arrived, our copies have arrived at opposite ends of the cell, and we can see here our DNA begins or uncondensing. We have a nucleus forming here, a nucleus forming here. We're still inside one cell, but we're about ready to split. And that brings us to our last part, which is actually the end of cell division, end of everything, cytokinesis, splitting. We're actually going to move our DNA to opposite ends and split into two different cells. Now, it's to be fair, we've got to be careful here. In animal cells, the cytoplasm pinches in, and we actually split into two new cells. The cell membrane pinches in, that's easy. In plant cells, a little bit different. Remember, they have a hard cell wall, so what has to happen is they actually have vesicles form and fuse at the middle to grow a new cell wall.